Welcome to the GeneArt Services Dashboard, where you can easily design and order all your custom gene synthesis projects, whether you want them delivered as clones or DNA fragments. Once you have placed online orders here, this is also where you will be able to monitor the status of your order in progress, review past orders, and even reorder plasmid preps for plasmids we have created for you in the past. To demonstrate the features of the powerful GeneArt Instant Designer application, we will take a cloned gene project as an example. When starting a new project, you may find it helpful to give it a name and a description to make it easier to locate later. If you choose not to provide a name now, the unique project ID will be used, but you can always change this name later. There are a couple of different ways that you can input your sequences. You have options to upload a file using FASTA, GenBank, or Excel format, or you can manually enter your sequences using copy and paste. The upload option can be useful when you have multiple sequences to add, and there is a link to download a template if needed. On the upload screen, simply choose a file or drag and drop it into the upload window. Once the file is ready, you can click Continue. In this example, one of the sequences must be reviewed before importing. On the pre-import screen, the uploaded sequences are each shown on a separate line, along with some basic information and default configurations. There is a red error symbol in the status column indicating one of the sequences requires further attention before continuing. Click on the pencil icon to take a closer look. The red bar indicates that the problem is with the host organism. When a protein sequence is uploaded, the host organism is required so the tool can select optimal codons for the DNA that will be synthesized. From here, the clone can also be configured to be provided in a vector other than the default cloning vector. Several popular expression vectors are available. If you have onboarded a vector with us, it will also appear in this drop-down list. When you select a vector, you may also need to select insertion points. A larger plasmid prep, suitable for transfection, can also be selected. All the sequences now have a green check in the status column, so they can be imported and saved. Next, you will have a chance to review construct configurations, optimize each sequence for improved expression, and review the final order. For guidance on these steps, please see our video on configuring clones.